Hello and welcome everyone to this first podcast that covers day 20, chapter 20. In these three podcasts, we're going to talk about biotechnology and how our general knowledge of things like transcription and translation and replication and mutations has led us to the ability to harness some of that knowledge into providing tools that benefit us, both from a healthcare perspective perhaps, and also from a societal perspective, and of course from an environmental and ecological perspective. In this first podcast, we're going to talk about how we clone a gene and what we can do with that cloned gene. And then we're going to move on to the third objective, which it'll be our second podcast, but the third objective feeds nicely right after the first objective, which will involve talking about gene therapy. And then the second objective, which I would encourage you to watch last, will be on cloned organisms and stem cells. Okay, let's go and get started here with gene cloning. I thought it would be a good idea to talk about a specific disease as we move forward with these three objectives. And the disease I thought about using, and that we're going to use, is diabetes. So I'm going to write that down here, diabetes. And more specifically, diabetes type 1. This is also referred to as juvenile diabetes because typically you're born with this form of diabetes. It's not something you typically develop later in life as an adult. This diabetes type 1 is characterized as not being able to make your own insulin. So I'm going to write the word, a minus sign and then insulin. So somebody with diabetes type 1, they can't make their own insulin. Last week, when we talked about signaling, we discussed how insulin is taken up into the cell using a long-distance signaling pathway, the endocrine system. And I want to remind us real quick what is happening here under a healthy situation when somebody isn't diabetic. So I'm going to draw a blood cell here. I'm sorry, blood vessel, not a cell. A blood vessel. In this blood vessel, I'm going to draw a glucose molecule. I'm just going to put a G in it. It's circle here in the G. And then I'm going to draw a target tissue, say something like muscles. So let's draw a muscle fiber here. Something like this. And the blood vessel is going to come close to this here like so. And on this surface of this blood vessel, the muscle cell, there is a channel. I'm going to draw this channel like so. And when this glucose molecule comes along here, it can't get in because the channel hasn't opened yet. So how do we get this channel to open? Well, that's when the insulin comes into play. So I'm going to draw insulin as a triangle here. Like so. This is insulin. Just going to put a letter I there. What insulin will do is it will bind, well, I should say there is a cell receptor here on the muscle cell that can bind insulin. So insulin is going to come along here, and it's going to bind to this receptor. And when it does bind to the receptor, it changes this channel, and it causes this channel to open up. So I'm going to expand the channel here, this glucose channel, and as it does, because the insulin is bound there and it changes the shape of this channel, now insulin can come into the cell. Glucose can come into the cell. So now glucose is in the cell. It can only get in the cell if insulin binds here. So that's why insulin is really important, so that glucose can get inside of these tissues. Now, if you're born without being able to make insulin, that means the gene that codes for insulin isn't there, and so you don't make the protein insulin. So insulin can never bind here, and glucose can never get in. So let's draw a situation here where if we didn't make this. So if we didn't make insulin, so let's just put an X through it. It wouldn't be there, so it couldn't move, and it couldn't bind here. So now glucose would never be able to get inside the cell. It would never be able to get inside the cell because insulin is not binding to the receptor, expanding that glucose channel. So now glucose is unable to get inside the cell. It's blocked, and so it just stays in the blood system. Okay, so this explains what's happening in a healthy individual who produces insulin. That's how they get glucose into their cells. And then when you don't have insulin, as in the case in diabetes type 1, we can see how lacking insulin prevents glucose from getting into the cells that need it. Now, in the early days, say before 1920 and way back into the 1800s, when people understood that there was this disease called diabetes, the only way you really could treat it was by limiting their diet, because they knew if they ate a lot of sugar, they'd get sick. And then, say, in the early 1900s all the way up to like 1920, they started making some observations. They started realizing if you took, for instance, the pancreas out of a dog, that it would develop diabetes. And then some others did some research that if you took some extract from that pancreas and gave it back to the dog, that the dog was once again treated for diabetes. So this started leading to some observations. And then at some point, they realized that they could isolate certain fractions of that pancreas and produce and purify that insulin, and they could give that back to the dog initially. And then at some point, they started giving it to themselves. The scientists started injecting themselves with insulin. It's not something we would do today, but it's how science was done back in the day. And then they found some children that had diabetes, and then they started injecting them with this extract from the dogs. And lo and behold, the kids were effectively treated against the effects of diabetes. And so this led to mass production. Well, first of all, it led to a Nobel Prize for the scientists who discovered it. And then it led to mass production of insulin. And I believe one of the first companies that started producing it was down in Indianapolis, Eli Lilly. They made this by having cows and pigs. They would from the slaughterhouses, they would get their pancreases and they would produce the insulin that way. This turns out to be very expensive and not as cost effective, and you didn't get as much insulin as you needed. So it didn't take them long to realize that using these large organs from these cows and pigs was not the most effective way to do it. But they didn't really understand genetics at this time. This was the early 1920s and moving into the 1930s. We didn't realize a lot about DNA and what it was doing. We certainly didn't understand transcription. We didn't really know what a gene was so much at that time. But by 1978, we had learned a lot. And we could make human insulin in E. coli. And that's what we want to talk about today. How could we make human insulin in E. coli and use that to mass produce insulin and give it to the diabetics who needed it? So let's go to a new slide. Okay, so it was clear that a new way of mass producing insulin needed to occur, and a few discoveries happened that made it possible for us to mass produce insulin. Some of these discoveries included plasmids. So let's talk about that first. So we have our bacterium here, and the DNA in the bacterium is in the cytoplasm, the chromosomal DNA. But we also discovered there were these extra chromosomal pieces of DNA, circular, that were called plasmids. And these didn't have a lot of genes on them, but they did confer certain traits. And we knew, the bacteriologists knew if we took this plasmid and put it into a new E. coli, that the new E. coli would now have some of the traits of this plasmid. This bacteria here. And so we knew we could isolate this plasmid. So let's go ahead and isolate this plasmid, and let's put it here. And we'll come back to that in just a moment. Another thing that we discovered were things called restriction enzymes. So let's write restriction enzymes. And in the future, I'm just going to abbreviate this as RE. So just RE. These are like enzymes that we've talked about before. They have a very specific action, and there are hundreds of these restriction enzymes. And what we know restriction enzymes do is they will cut DNA. So if we put a restriction enzyme with this plasmid, it would cut this plasmid, and it wouldn't degrade it. It cuts it at very specific sequences. We'll come back to that in a moment. Another thing we learned how to do was to identify genes. And sometime in the 1970s, we were able to identify the insulin gene, the human insulin gene. We were successful, and when I say we, of course, I don't mean myself, I mean the scientists of the day, were successful at finding this gene in humans. And so they knew which gene made the protein insulin. But being in humans, it was really tough to do anything with it because you couldn't mass produce insulin in humans and then somehow or another extract it from the humans because obviously that would be a very unethical thing to do. So we want to avoid that. But how could we mix these together? Okay, so now there's one other thing we have to identify that we discovered, or the scientists discovered, and that is DNA ligase. And we've talked about DNA ligase before and its role in replication, and it's the same DNA ligase, but we were able to purify it and use it. We'll talk about how we're going to come back and use that in a moment. All right, so we have our plasmid here. And we know in theory, they thought we could put the insulin gene in this plasmid and then put this plasmid back into the bacteria. Back into bacteria. Then the bacteria would use its own systems for transcription and translation and make human insulin. And then we could give that to people. Sounded like a great idea. So they take these restriction enzymes and they mix it with this plasmid. And I'm going to draw this plasmid as a double-stranded because it is double-stranded. I just didn't draw it that way before. And one thing it does, let me just draw it first. And I intentionally drew it this way, where this gap here. These are appropriately called sticky ends. So let's just write that word here because I think that's on your study guide. And again, remember, this occurred because we treated this original plasmid here with these restriction enzymes. We added them to this plasmid here, it cut them, and we ended up with this cut circular DNA. When it cuts it, it always leaves these sticky ends. Now, somehow or another, we had to get insulin and put it in here. Well, initially, they could take this insulin from the human genome, they isolated human DNA, and they were able to insert it in there. And we'll talk about how they did that in a moment. But I'm going to jump ahead and talk a little bit about how we do it today because we have better technology today. And we can take insulin, this insulin right here, and mass produce it, just the gene, not the protein, just the gene, so that we have millions of copies of the gene. Okay, and I'm just going to draw lots of copies here. Well, I'll just draw 3, but it can make millions of them, right? And we do this by a technology called PCR. PCR stands for polymerase chain reaction. The short story is it will amplify this DNA over and over and over again. Now we have lots of copies of this DNA, and we can engineer them so that it reacts with the same restriction enzyme. So we can take this insulin gene and add the restriction enzymes to them, and what they will do is they will cut these as well. Now, I drew these as a single line, but it's DNA, so it's going to be a double helix. There's going to be 2 strands of it. So I'm going to redraw one of these double strands over here. And if you notice, this restriction enzyme cuts it in the exact same way, so we still get these sticky ends. And if you'll notice, this sticky end of the insulin gene and this sticky end of the plasmid match each other. So when you mix them together, this will land right here, this in here. In fact, why don't we just draw it on here? It'll land just like this, and it's right there in the plasmid. Now we need to finish this off because see how I purposely did not connect them here? What has to happen now is our friend over here, DNA ligase, comes into the picture. And DNA ligase, as the same enzyme that our cells use every day to copy our DNA and to ligate the ends together here, comes along here and it fills this gap here and connects them. And so now what we end up with, I'm going to redraw this here, is a successfully cloned human insulin gene in a bacterial plasmid. So here's our plasmid, and I guess I'll still draw it in this kind of with 2 strands here, just so we remember that it's DNA, so there are 2 strands to it always. Even though for simplicity, I didn't draw it initially, but there are 2 strands here. So this is still plasmid DNA containing our insulin gene. All right, so now we have the plasmid. We're almost there. This plasmid with insulin in it. Okay, now what we do is we take this plasmid and we want to put it back into the bacterium. So here's our bacterium. Please excuse the size difference. Obviously, this plasmid is much smaller than the bacterium. But we're going to take this plasmid and transform it, and place it into this plasmid into this bacteria. So this plasmid is taken and placed right back into the bacteria. And now it's in the bacteria. So let's draw it again. And now I'm going to just draw it much simpler because of our space requirement here. We have this plasmid with the insulin gene now in the bacteria. And now what we can do is the thing we've been waiting to do, and that is to place this bacteria into a large flask. And in industry now, they'll use these large vats that hold, maybe 500 gallons of media. So we put media in here. We take our bacteria containing our plasmid with our insulin, and we place it in here, and we let it grow. Bacteria grow super fast. They have a generation time of every 20 minutes. All the time, every time they divide, they make more and more insulin. So now what you do is, after a certain amount of time, not long, maybe a day or so, you take the cells out of here, you bust them open, and isolate the insulin protein. So this is a good opportunity to review some of the things that we've learned about before. This gene, it happens to be a human gene in a bacterium, but because humans and bacteria genetically, we're really not that different. And the way we run our cells is nearly identical. This gene is going to be transcribed into messenger RNA, and that messenger RNA is now going to be converted into, translated into insulin protein. So now you have this big vat of E. coli growing, just making more and more and more insulin all the time. And so now you just take this liquid part here, take it out, and we isolate our insulin protein. Not the gene, but the protein. So this, and I'm just going to kind of draw this squiggle mess because it's going to be fully folded. And in this way, we don't have to take these large organs from cows or pigs. We just need a much smaller room where we can grow lots of media and mass produce these proteins. And now we can take this protein and put it in, a bottle of some sort, and now the person with diabetes can take this medicine daily and inject it in themselves and survive. And it's much cheaper, it's much more efficient, and another big advantage is what we're putting in humans right now isn't cow insulin or dog insulin or pig insulin, it's human insulin. So now we at least have the right species of protein being placed into the human. And it's very effective. So if you know somebody with diabetes type 1, or if you by chance have it yourself, this is how your insulin is made, or your friend's insulin is made. It's mass produced in bacteria. Today, a lot of times I think they might use yeast, but it's mass produced in a microorganism. And we know this. I always like to kind of go back to this. The reason we can do this is because we understood the basic biology of it. We understood transcription. We understood translation. We understood replication. In fact, if we didn't understand replication, the scientists didn't understand it, they would have never ever identified DNA ligase up here. And without DNA ligase, this really important enzyme in replicating our chromosomes couldn't be used here, and we wouldn't be able to mass produce insulin. So basic research is so important in understanding and implementing therapies for our life. Okay, well, I hope that made sense. That's the end of Objective 1. And the next podcast is going to be on Objective 3. And in Objective 3, we're going to talk about gene therapy. And it's going to be very similar to this, where we're going to create insulin in a foreign piece of DNA, but instead of taking the protein from a bacteria and putting it into the human, we're going to put the gene into humans. All right, so you have to tune into the next one to learn a little bit more about that. If you have any questions, let me know. Thanks. Bye.